Introduce Sean O'Connell, PhD. He's founder and president of O'Connell Medical Consultants. He has a background in immunology, cell biology, regenerative medicine, and tissue repair. And he's an adjunct professor in the Department of Vascular Surgery at Englewood Hospital and Medical Center. And when I had a conversation with Sean back in February, he told me that he has met Dr. Fauci, and he said that Dr. Fauci is his hero,
Yes.
in this pandemic, and I guess before and any time. So Sean, it's all yours, my friend.
Thank you
Thank
so
you
much
so
for
much.
that wonderful introduction.
Can I just, before we start, can I just make sure that we're recording this? Pastor, you're going to record it for us?
Yeah, it's being recorded now.
Okay, thank you.
Good. Well, good morning, everyone, and thank you so much, John, for that nice introduction. What I'm going to tell you about today clearly is about the COVID vaccines. We were just talking about them, and there's a lot to know about the vaccines. This is not a simple thing. Pastor, if it's possible, do you think you could do some selective muting until I'm done? Because that way I can just go through it, and then we can open up everyone's mics and take questions.
Sure. Yeah.
I think that would be the best. All right. So today I'm going to talk about the COVID.
Sean's muted.
Sean, can you please unmute yourself? Because
How about that?
yeah, sorry about that.
Very good. Yeah, you have to do that selectively. So today I'm going to go through the major vaccines, how they were made, and focus on the ones that are available here in the United States. I'll have some words about some of the other vaccines that are coming on, but I want to concentrate on the ones that are currently available in the US. All right, let's see if we can get this to go. There we are. All right, so the first topic is going to be, what are the COVID-19 vaccines? What's currently available? And by available, I mean those that have received this emergency use authorization, and what's coming down the pike soon after that. I'm going to talk a little bit about their development and the types of vaccines, and then how do they differ from each other? How do they differ from conventional vaccines? And of course, how are they tested? And by this, we're talking about how is their efficacy, their effectiveness determined, and how is their safety determined? All right. We're also going to cover the corona variants. These have popped up within the last couple of months, and they are of concern. And I'm going to go over what currently is known about the variants that are currently circulated in the United States. Talk a little bit about vaccine distribution. That changes on a daily basis. So I'll give you what I have as of this week. And as I mentioned before, I'm going to finish with a more detailed description of the side effects and adverse reactions that have been associated with these vaccines, and how to protect against that, how to plan against that. So how do these vaccines work? To answer that, we have to ask, what do we mean by vaccination? Well, I'm going to talk about that in just a minute. How are they developed? We have nucleic acid vaccines, we have viral vector vaccines, protein adjuvant vaccines. This gets us into something that's new in vaccine technology called the cassette model, or the cassette platform, and I'll talk about that. And then what is their efficacy against the original coronavirus, as well as the new variants? All right, so what is vaccination? Administration of a vaccination is done in order to stimulate a person's immune system to protect against a particular disease. That's usually an infectious virus or other organism, bacteria. Traditional vaccines, going back to the original smallpox vaccine and polio vaccine of our youth, were made from weakened versions of the virus or killed versions of the virus. They were then later extended to just include proteins that were taken from the virus. None of the current, and I'll say this right now, none of the current vaccines that we're going to talk about today are either killed or attenuated live vaccine viruses. None of those are being circulated in the United States. Why is that important? You cannot catch this disease from taking any of the vaccines that are currently approved in the United States. Vaccines can be given in either one dose or in multiple doses. Multiple doses generally serve to better stimulate the immune system. They're usually by injection, but there are oral and nasal vaccines out there. What do they do? These vaccines basically stimulate the immune system by recruiting specific cells within the body, called memory cells. These cells are specific to that organism alone, and the markers on that organism, and they give rise to both, as I show in the next bullet, antibodies that can recognize and bind to the, in this case, the virus, the COVID virus, or they make specific cells, T cells, that will kill virally infected patient cells. So they'll recognize and destroy them. And as we've heard much in the news, when sufficiently large percentage of our population gets immunized and has immunity, we achieve what we call herd immunity, which means that there's enough people that have effective immunity that the virus can no longer spread, can no longer get a foothold. That's what we're all shooting for with these vaccinations. All right, so what are some of the types of vaccines we're talking about? Well, the first and most novel and the most common around here are the genetic material vaccines. These are mostly RNA vaccines, but there are a couple of DNA vaccines that are currently under development. One of them is in late stage. But around here, what we mean is, by this, is the Pfizer vaccine or the Moderna vaccine. These are made of a piece of RNA that represents part of the virus. And what happens is when they get in the cell, this RNA is converted to protein that's specific to the virus. That protein shows up on the surface of the cell, and our immune systems can then see it. All right. Another type of virus, which is represented in the US by the J&J virus, are the viral vector vaccines. In this case, we use a benign virus, in this case, an adenovirus, that's specifically engineered to just be a carrier for the message, in this case, associated with COVID, what we want to immunize the host with. And essentially what we do then is we put a small amount of this genetic material, in this case in DNA form, into this adenocarrier. That gets injected, and what happens then is the cells then make the RNA from the DNA and protein from the RNA, and again you get expression of the viral proteins on the surfaces of some cells so that our immune system can see it. The J&J is the most common. The AstraZeneca is also common. So is the Russian Sputnik vaccine. These are all viral vector vaccines. All right. Finally, they're the protein-based vaccines. And this is important. There's a vaccine that was developed in Maryland called Novavax. Here, what they have done is they take proteins that are associated with the virus. They make a recombinant DNA form of this piece of protein, and they generally mix it with molecules called adjuvants that further stimulate the immune system. In this case, these adjuvants are nanoparticles, and you get injected with that. This is also seen, for example, if you've got any of the newer flu viruses and antivirus vaccines, like the Flu Plus. They use recombinant proteins as the basis of the vaccine. And this Novavax is scheduled for approval within the next couple of weeks for emergency use. And it's a good one too. It's a protein vaccine. But again, you cannot get the disease from any of these. They only represent fragments of the COVID virus. And then finally, as I mentioned earlier, they're inactivated or attenuated viral vaccines. These are common outside the United States, but none of them have been approved and none of them are being distributed in the United States. The issue with them is that, yes, you have to make sure that your inactivation is effective, and they don't seem to be as potent. The highest I've seen the efficacy is about 50%. This is compared to the 70 to 90+% that the other vaccines I've shown you can achieve. All right, so this is an important thing. How are they tested? I get a lot of people asking me, well, hasn't this been rushed? didn't we do this too fast? Don't we need more time? Yes, the development was done quickly, but no corners were cut. There is a standard vaccine testing process, which I have here on this somewhat busy slide. And I'll just give you the highlights. You start in preclinical testing. You have a candidate vaccine that you think is going to work. The first thing you do is you test it on cells and see if it protects cells in the petri dish. And then you move to animals. And the first thing you ask in the animals is, well, does it induce an immune response against, in this case, the virus? And then finally, can it protect that animal if you challenge it with the virus? All the vaccines have gone through preclinical testing. Then you go to phase 1 safety trials. Researchers give the vaccine to a small number of people and they test for safety and appropriate dosage. The dosage can change with the vaccine. And again, you're looking for, is it safe and does it stimulate the immune system? If the answer is yes, you go on to phase 2. This is expanded trials. You now give the vaccine to hundreds of patients, and those patients are split into groups like the elderly or children 12 to 16, which they're doing right now in the United States with the Pfizer vaccine. You want to see if the vaccine acts differently in these different groups, and you further explore safety. And then finally, you go into phase 3 trials, which are efficacy trials. Now the vaccine is given to 1,000 people at different centers. And what you do in a vaccine trial is you have a control group and you have a test group. They both get either a placebo in the control group or they get the actual vaccine in the test group. No, they don't know, neither the doctors know which is which, the patients don't know which they're receiving. And then after you give that, either the placebo or the test, you ask, all right, going forward, how many people get the disease? And this is what determines efficacy. That ratio is your efficacy. So if only 10% of the patients get the disease in the treatment group and a larger number get it in the control group, you can say that vaccine's 90% effective because it protects 90% of the people receiving it from getting the disease. This is why historically vaccine trials generally take a long time. Why it's been able to be done so fast, not only for the reasons of the money that was spent and the resources that were obtained, is the fact that we had a lot of disease in the United States. We've had a ton. I can't overestimate this more. There's been so much disease in the United States that waiting for enough people to get the disease wasn't a problem. So we were able to get the phase 3 trials done in a relatively short fashion, such that when these vaccines first started to gain their approval last December, we had more than adequate numbers of patients to fulfill the phase 3 requirements. Since then, we've obviously gone from tens of thousands of patients to now millions of patients. So we have a pretty good idea now of the efficacy and of the safety of these vaccines. All right, what happens after phase 3? Well, as I just mentioned, many countries, based on the early phase 3 data, will give emergency use authorization. And they're basically saying that for emergency use, they are safe and effective. In the case of China, Russia, and some other countries, they gave the emergency authorization early after phase 2. In the US and the UK and in Europe, they were much more conservative. Emergency use was only given after a significant amount of the phase 3 data was made available. Approval, and you'll hear this, approval takes a lot longer. Approval is when you finally finish all your phase 3 trials, you've locked your data set so that it's there. Then the regulatory agencies look at it and say, all right, yeah, this is a good vaccine. We're going to approve this for continuous use. And it's important to note that right now in the US, none of the vaccines have full approval. They all have, the ones we're talking about have emergency use authorization. There's no doubt that they will gain full approval, but they don't have it right now. And then finally, what happens after this is there's always post-clearance, post-regulatory approval surveillance. they always monitor vaccines to see if there are any side effects that you can't see when you're treating tens of thousands of patients or hundreds of thousands of patients. They only show up until you're getting into the millions. And so it's always important to do post-approval surveillance of vaccines for both safety and efficacy. And as I said before, all vaccines that we are talking about today that have achieved emergency use authorization have gone through all three phases. So there was no shortcutting. There was no corners that were cut. That's very important. All right. So what vaccines are we talking about today? Well, around here in Morris County, the most common is the Pfizer vaccine. It was developed between Pfizer and a German biotech company called BioNTech. And it's an RNA virus. It's the one you will see if you go to the Rockaway Administration Center. And it has emergency use authorization. There's also the Moderna, a very similar mRNA virus developed in New York. It has EUA. You can see that around here. AstraZeneca is a viral vector vaccine that was developed between Oxford University and the British Dutch company AstraZeneca. It's awaiting emergency use authorization. There's still the FDA is still waiting for more data on AstraZeneca. Johnson & Johnson, that's a single dose viral vector vaccine that has been given emergency authorization. I'll talk about the advantages in just a minute, but it's a single dose. And then, as I mentioned, Novavax is a protein adjuvant vaccine developed in Maryland. That's going to probably receive EUA within the next month. But it's important to note there are another over 144 vaccines currently developed worldwide. So we won't have a going forward, there won't be a supply problem. There'll certainly be vaccines to choose from, but right now, of course, everyone is keen to get immunized. All right, let's now talk very briefly about each one of these vaccines in a little more detail. The Pfizer vaccine is an mRNA vaccine. It requires 2 doses spread 21 days apart. It's got emergency clearance. As I said, it's an mRNA virus. So what happens is the virus enters the cell, like the muscle cell. You get it injected in your arm. And the muscle cells take up this mRNA. And from the RNA, you make a portion of the spike protein of the COVID virus, which is then expressed on the cell surface. The immune system can then see it and respond to it. It's good for in adults. It was tested initially in adults 16 years and older. It produces both antibodies against the virus as well as these immune T cells. In the original trials against the parent strain of COVID, it was 95% effective after the second dose. There are similar results. It has similar results against this UK strain, slightly lower efficacy against either the Brazilian or the South African strain. This is based only on bench tests, not on actual tests out in the field. They're doing that now. It has been tested in over 40,000 patients in the pivotal trials, the phase 3 trials, and it's currently being studied in children 12 to 15. They're trying to extend it down to the younger patients who were not involved in the original trials. There were no significant side effects noted in the phase 3 trials. You had, the common ones like maybe headache, maybe chills. You could have maybe a fever lasting a day or so, but really no major severe side effects were noted in the phase 3 trials. I'll talk a little bit more about this in a minute. One of the problems with the Pfizer as well as the Moderna is they really need cold storage. In the case of the Pfizer, ultra cold. It needs to be stored at about minus 70 degrees C, minus 94 degrees Fahrenheit. That's pretty cold. So you need specialized freezers, and you need these specialized shippers using dry ice. The Moderna is a similar vaccine. It needs 2 doses. The only difference is it's 28 days apart instead of 21 days apart. It has clearance in the United States. Again, it's an mRNA virus. You administer it through muscle injection, and they used it in adults 18 years and older in their trials. And it was shown to be 4.5% effective against the parent COVID strain. This is measured 7 days after the second dose. And it too has similar results against the United Kingdom, the B117 strain. It was tested in about a little over 30,000 patients, 18 or older. And it's currently has 2 studies ongoing, one in very young children, 6 months to 12 years, and then a second study in children 12 to 17 years. These studies are currently ongoing. The whole idea is they want to expand the population that can get this vaccine. It's a little better on storage. It has a shelf life of 30 days in the refrigerator or 6 months in a standard freezer, and it'd be -4 degrees Fahrenheit. Then there is the AstraZeneca. This was supposed to be the big world blockbuster. It's a cheaper to make virus. In the pivotal trials, 2 doses, 12 weeks apart in patients 18 years or older, gave about over 82% effectiveness. It was made using a specialized adenovirus developed at Oxford University. It's administered by muscle injection. However, while it is effective against the parent strain of COVID, it is not effective against the South African strain, nor is its efficaciousness decent against the Brazilian strain. It was tested in about 32,000 patients. They have a submission for emergency use pending with the FDA, and they're expecting that one to be approved at the end of this month, but so far it hasn't. It's also well tolerated, no severe adverse events in any age group. However, they didn't treat as many older people as the other vaccine studies did. One of the big advantages besides lower costs is it's stable in the refrigerator for about 6 months. So it's easy to transport, it's easy to store. Then there's the Janssen & Janssen drug. This was just recently approved, I think like a week or so ago. It's a 1-dose drug, though they do have a 2-dose trial ongoing. But it's a 1-dose drug. It's a viral vector like the AstraZeneca, uses a virus called adeno-26. And again, it's administered by muscle injection, adults 18 years or older. It's got about 72% efficacy against COVID, the original COVID. But one of the things to note, in fact, for all of them, is they all are effective in preventing severe disease and death. They all have a very beneficial effect, even if there are some differences in their absolute efficacy in preventing infection. They all are very good at preventing severe infection. Again, they have similar results against the UK strain, and they're also fairly effective against either the South African or the Brazilian. They've been testing about 45,000 plus patients, and you can store them for up to 2 years in a regular freezer. All right, and then finally, Novavax. It's the one I mentioned. It's the new kid on the block. It's from a company in Maryland. It's a protein. It's not a nucleic acid. It's not a viral vector. It's a protein vaccine that is the proteins are embedded in nanoparticles, and they serve to induce the immune response. It's administered in 2 doses, 3 weeks apart, and was tested in people 18 to 84, muscle injection, and it's quite effective. It's 90% against the parent strain and against the UK strain. It's still fairly effective against the South African strain. It was tested in South Africa, and it's stable in the refrigerator. We're expecting an emergency use clearance on this vaccine by early mid-April. All right, so as I mentioned before, they were developed quickly, but they were not, no corners were cut. Nothing was short shrifted. And the question is, well, how could they do that? Well, the fact of the matter is that while vaccines normally take years to produce and approve, for example, up until COVID, the fastest vaccine approval was for the mumps vaccine, the part of the measles, mumps, MMR, measles, mumps, and rubella. That took 4 years, but that's a conventional vaccine. You're basically taking virus and you're killing it with heat or chemicals, and you're incorporating it with adjuvant, and you're testing it in a wide group, in this case of kids for the most part. The vaccines we're talking about today are all quite modern, but they've been all under development for decades. The RNA vaccines were developed starting about 20 years ago, the BioNTech ones. The same with the AstraZeneca that was developed in Oxford originally about 25 years ago. And they were used predominantly to make vaccines against really horrific infections like Ebola, Zika, dengue, and HIV. And so they have been around for development for quite a while. One of the big things about this, and I mentioned this earlier, is they form kind of a cassette model. What do I mean? If you have the DNA sequence or the RNA sequence of your virus, you can take that sequence, you look for the parts of the virus that are most likely to raise an effective immune response. You take those sequences and you plug it into your cassette, and out comes your vaccine. When the when the RNA sequence of the COVID vaccine was published in January 15th, 2020, within 4 days, Sarah Gilbert at Oxford University had a working version of a vaccine that would later become the AstraZeneca. The BioNTech and the J&J people quickly followed. So it was just a matter of once that sequence was available, a working version of these vaccines was rapidly developed. It was sort of waiting to happen. And then all it needed to do was the testing, which I mentioned earlier. You had to go through the 4 phases of the testing. This new development in vaccine technologies is extremely important because we know that these vaccines mutate quickly. In a manner like influenza, COVID changes its genetic structure. The way it reproduces is error prone. That is, it makes mistakes. The more it has cells to infect, the more virus it makes, the more mutations can occur, the more variants are going to show up. And I'm going to talk about more of that in a minute. So why this new method of making vaccines is important is that vaccine makers can then respond to these mutants very quickly. In fact, it takes about 6 weeks in total, on average, that if you have the sequence of the variants, which I'm going to show you, you can make a new vaccine. And all the manufacturers already have what they call booster vaccines in development, being tested for the new variants. We never could do this before. We always had to go back from scratch. And we all remember that every year we have to say, all right, what kind of vaccine coverage do we have from influenza? How good is it? And this is important. The best influenza ever did, the best strain of vaccines was about 50-some% effective. Typically, it could go down to the 20s, depending on the year and the strains involved. These vaccines are all in the 70s, 80s, and 90s. So this technology clearly is superior to the old technology, and it's much quicker and easier to develop new vaccines as the new strains emerge. I can't emphasize this point more strongly. All right, so let's talk about this. Let's talk about the variants for a second and how the vaccines work on them. This is a diagram of the genetic information, the genetic map of the parent COVID strain, SARS-CoV-2. So you have we depict these things as circles. This is the RNA, and it shows the proteins that the RNA is going to have the instructions for. And this RNA is packaged in the viral capsid, which you see in the middle. That's that circuit with the little spikes, the spike proteins coming out. Then the spikes are attached to a protein coat, a capsid. And then within the capsid, you have your RNA. All right, this virus codes for about 30,000 letters of RNA. And it takes 3 letters to make an amino acid, and it takes about a couple of hundred amino acids to make a protein. So there's about 20 to 30 some proteins that are in theory coded for this virus, of which about a half a dozen or so are important. The most important for our purposes is the spike protein, because the spike protein is what allows the virus to attach to a cell, get into that cell, and then turn the cell's mechanisms, machinery into making new viruses. In the case of the RNA viruses, they make a lot of mistakes in this process. And these mistakes are mutations. There's changes in the RNA code. And more mistakes are made by these RNA viruses than other viruses because they don't have nearly as efficient mechanism, for example, that our cells do, of checking itself as we copy, as a cell divides and copies its DNA and you make new cells. We have very efficient mechanisms within our cells to make sure we don't make mistakes. These viruses don't. They make a lot of mistakes. And the more they can replicate, the more they can produce more viruses, the more variants. This is why it's so important to achieve herd immunity, even if you don't have the new vaccines for the new strains. If you can stop transmission, you're going to stop viral replication. You're going to stop the emergence of new variants, or at least slow it down considerably. So that's why right now there's a huge push to get as many vaccines in as many arms as possible. All right. So what about these mutations? Well, as they accumulate, certain of these mutations clearly are strong. Many of them don't have any effect. Many of these mutations within the virus are bad and the virus dies. Some of them confer a benefit to the virus. And in our case, we're interested in two things: mutations that either allow the virus to be more contagious, to bind to more cells, bind to those cells tightly, and hence infect more people. The other one is that they cause more severe disease. And some of the hot variants that are out there, you see both types of mutations. Mutations that make them more contagious and spread quicker, and mutations that give rise to more severe disease. If you have enough groups of viruses that share the same mutation, we call that a variant. And if that variant becomes stable, we call it a strain. The original COVID virus that apparently came out of China in the Wuhan region is a strain called SARS-CoV-2. This is different, for example, than the SARS back in the early 2000s that caused the acute respiratory distress syndrome, or the MERS that came out of the Middle East that was also a type of coronavirus. There are coronaviruses that cause common colds. Those are all different strains. However, within the strain of COVID now, SARS-CoV-2, we have different variants. And these are what are emerging that are giving cause to a lot of concern. These are the current efficacies that you got from the vaccines that I've talked about: the Pfizer and Moderna, the Johnson & Johnson, AstraZeneca, and Novavax. When you go to other strains, those numbers are going to decrease. The first strain of any note that was important, the variant from UK called 117, spreads a lot faster. And right now it's going to be, certainly probably by the end of this month, the dominant strain within the United States. It's a very rapidly producing, rapidly spreading strain of COVID. The good news is most of the vaccines we're talking about are still effective. They're about over 75% effective, as opposed to 90%. It's good that we have effectiveness because, as I said, they're doubling every 10 days in the United States, and they are rapidly becoming the dominant strain here. One of the bad news is a recent report in Nature showed that they do have a higher incidence of severe disease and death compared to the parent COVID strain, about 55% higher. More troubling are the two strains that I've mentioned, one out of South Africa and one out of Brazil. For short, we call the South African strain 351 and the Brazilian strain P2. They share multiple mutations. And the big mutation and the big problem with it is that they cause more severe disease. Also, they are much less susceptible to the vaccines, to some of the vaccines. And as a result, all the vaccine companies have developed now boosters for these two strains. But the vaccines that we're talking about, in particular the Pfizer and the Moderna, are still pretty effective against these. And they seem to be able to completely block severe disease and death. But they might not be as effective as preventing you from catching it at all. And I've got the numbers there. The Novavax about 55% effective, the J&J about 57%, but the AstraZeneca is poor against these strains. The good news is, as I said, all the vaccines seem to be able to stop severe disease and hospitalizations and death as a result of these new variants that are of concern and currently circulating. All right. I've already mentioned this. Because we have cassette platforms, which each one of these vaccines, boosters against the particular variants are currently under development. They take about 6 to 8 weeks, and right now they're in safety and efficacy testing on all the companies concerned. This will be important going forward because one of the questions that people ask is, well, how long does the immunity last? And the answer is we don't really know. We do know that it lasts longer than people who just get a mild to moderate disease itself. That immunity doesn't appear to last terribly long, maybe 6 to 12 months. The vaccines confer a longer lasting immunity. In addition, if you've had COVID and you get a single vaccine dose, that gives you a very strong, apparently reasonably long lasting immunity that can last, looks like at least a couple of years. But sometime, unfortunately, as with influenza, hopefully not as frequently, we will need revaccinations in the years to come. How frequent that is, it's impossible to tell at this time. Another big question that people ask is, do these vaccines block transmission? It's a huge question. The answer is the jury is still out. It's not known for sure. However, preliminary studies in Europe and in the US are encouraging that it can drastically slow down transmission within several weeks after your second dose, but we don't know for sure. We don't have really good numbers on that. So that's why it's still important that after you've received your vaccine, your second dose of the Pfizer, for example, you still need to wear a mask going out in public. You still need to social distance until most people become immunized, the herd immunity. I'll say more about this. I get a lot of questions. If you got a group of people that are all immunized, obviously this isn't a problem. If you're immunized and you're with your own family and you know the situation there well, you can probably ease up on masking and social distancing there. However, if you go out in public or you're with a group of people who you don't know what their status is, it's still a good idea to mask and it's still a good idea to social distance. And it will be until, let's say, later this summer. All right, what about the rollout of the vaccine? All right, as of today, about 118 million doses have been delivered in the United States, at least one dose. That represents about 23% of the population. We're going at a clip now of about 2.5 million, somewhere between 2 and 3 million a day. This is a far cry from January, where we were less than 1 million a day, 100,000. At this rate, now this is important, we have to be careful how we say this. At this rate, 50% of the US population will be vaccinated by mid-May and 75% by June, July. This is as opposed to what, for example, is being said by the current administration, where we're going to have enough vaccine for everybody in May. But that's, I say down there in a lower bullet, that's the distinction between vaccines and vaccinations. Having the vaccine is one thing, getting them in people's arms is another. Currently in New Jersey, we've got about 24% have received one dose, with 13% fully immunized. That represents, the good news is we're using our doses as we get them in New Jersey. The demand is still high. We get doses in from the manufacturers, we use them almost immediately. We're like working about 80% utility. So we're not stockpiling a lot of vaccine here in New Jersey. Why has the rollout been slow? Well, initially there were manufacturing logistical challenges. There was a need to have centralized plan for distribution. It's government coordinated. This is now been drastically improved, and the rate of vaccination is sped up tremendously within the last month. And if you look at it, the states are now expanding. That we, at first, you'll see this in the next slide. At first, we were like 65 and older. Originally 75 and older. Now it's 65 and older. Now that's coming down in most states. But then the demand still outstrips the supply. The administration initially pledged 100 million shots by 100 days in office. We're going to far past that, but it's still not fast enough. The current estimates on herd immunity, which is 70 to 90%, aren't going to occur until the summer. So we need to step it up. But this is where the authorization of the J&J shot vaccine, which is one shot, and the Novavax are going to help immensely. They're going to speed this up. Moving forward, these are the original guidelines about who gets it. And the problem with this is they change every day. It had a big change in December. It has another big change within the last 2 weeks. Now, on average, it's people 60 and older are eligible, and anybody below that who have a comorbid condition that is associated with more severe disease, they can get it, as well as all frontline medical workers, essential personnel, which is expanding. It differs from state to state. For example, in New Jersey, up until recently, teachers and grocery workers were not considered essential. Go figure. I think that's been changed now. As I said, this changes on a daily basis. What do we mean by comorbid factors? This is actually important. If you've had cancer or have cancer, if you have kidney disease, if you have COPD, acute chronic emphysema, if you have Down syndrome, if you have any serious heart conditions, heart failure, coronary artery disease, any cardiomyopathies, if you are obese, in particular, if you're severely obese, if you have sickle cell, or if you smoke, or if you're like myself, a type 2 diabetes, you have a comorbid condition, and that allows you to get the vaccine at any time now. You should just, and to get the vaccine, my suggestion is not only sign up with the state, sign up with the local health department. In this case, Atlantic Health is our biggest one around here, and sign up with the individual hospital organizations so that you're on all of those lists. So as soon as their computer matches you, boom, they give you an opportunity to make an appointment, you make the appointment. And I find you have to shop around. Now we're starting to see the vaccine distributed in addition to large mega sites like at the Rockaway Mall. We're starting to see this in CVS pharmacies. So there are multiple outlets, and my advice is sign up for them all. All right. Finally, I'm going to talk a little bit about the side effects and adverse reactions. This is important. We're going to get vaccinated. All right. The first thing we have to make a distinction is between simple adverse events that happen with any vaccination and severe adverse events. Severe adverse events with these vaccines are very rare. In the case of Pfizer, it's about 6 in a million doses. In the case of Moderna, about 2 in a million. With the others vaccines, it's even less. And by severe adverse events, I'm talking about anaphylaxis, a severe allergic reaction, rapid onset of symptoms. It starts off with itchy rash, throat and tongue swelling, shortness of breath, lightheadedness, vomiting. It's bad. Just like a bee sting or a peanut allergy. The good news is it responds very quickly to things like epinephrine. So like EpiPen can quickly resolve it, but it's serious. And as I said, this kind of adverse event happens just, a couple of times in a million doses. It's not common. Guillain-Barré is a neurologic disorder associated with some vaccines in the past. For example, most notoriously, the 1976 swine flu. So far, Guillain-Barré has not been seen with the COVID vaccines, which is a good thing. If you have a history of Guillain-Barré, definitely you have to tell the people, consult with your clinicians, consult with your doctors, and consult with the clinic administering the vaccine. Let them know ahead of time if your doctors give you the go ahead. But so far, knock on wood, there have been no real associations that I've seen anywhere with Guillain-Barré syndrome and the vaccines we're talking about. If you do have a history of anaphylaxis, like bee sting allergies or severe food allergies, you need to let the clinic know ahead of time. And the CDC guidelines are, instead of waiting 15 minutes, wait 30 minutes, just to make sure that you're okay. Because if you do have a severe reaction, as I said, shows up very fast, shows up within minutes. So if you hang out for 30 minutes, that's generally pretty good. And of course, the clinic should be able to handle a severe reaction if it occurs. So that's another thing that's good to check. For example, at the Rockaway, they're very good at the mega site. They're very good at checking, and they have stations to handle such reactions. It's on the second floor. All right, so that's pretty much the meat of my main presentation. What I want to do now is see, we've been there about 45 minutes. I'd like to spend the rest of the time basically addressing any questions you might have. And again, I thank you so much for affording me your time for me to make this presentation to you.
I have a question.
Yes.
If you can explain what herd immunity means?
It's a very good question. At some point, enough people in a population become immune to a particular disease such that that disease no longer has enough victims, if you will, potential victims to spread. Herd immunity can occur naturally. For example, in the worst case, the Black Death in the Middle Ages, it basically got everyone who was susceptible and left those who weren't susceptible anymore. And now the susceptibility in the population is very low. That's a gross example. A good example is polio. We basically eradicated polio through vaccination, first with the Salk and then the Sabin vaccines back in the 60s and early 70s. And herd immunity was achieved very quickly. More recently, herd immunity in measles was achieved, though some hesitancy, vaccine hesitancy, has allowed measles to creep back in. Another determinant of herd immunity is how efficient is the disease at transmission. If it's not very good at transmission, herd immunity is easy to achieve. If the virus in this case is good at transmission, and the poster child for this is measles. Measles is a highly contagious disease. Then it's harder to achieve herd immunity. It took us a number of years with measles, but we have achieved it. In the case of measles, one infected person can infect as many as 15 people. In the case of COVID, that reproduction rate, they call it, is about 2 to 3, especially with these newer strains. So in order to achieve herd immunity with a reproduction rate of the virus somewhere between 2 or 3, and maybe with the South African and British strains, maybe it's a little higher, it's been estimated at about 70 to 80% of the population needs to have immunity in order to achieve herd immunity, at which time the virus can no longer spread in that population. Carol, does that answer your question?
Yes, sir, it does. Thank you.
You're welcome.
Hi, Sean. Very good presentation, by the way. Thank you very much. My question is about herd immunity. Why is it, and I've heard this a lot from people, why is it we wouldn't want to wait for herd immunity to be acquired naturally as versus with this aggressive vaccine program?
You want the straight answer?
Sure.
I don't like people dying.
There you go.
We're up to 565,000 people. To me, this could have been prevented. That's all I'll say. Had we moved even faster, we could have prevented this more. I think we got the vaccines out as fast as we could. But the thing is, non-vaccine interventions, had they been applied uniformly and thoroughly, could have reduced this. So now that we have the vaccine, we have a solution. I don't think there is any defendable reason not to pursue aggressive vaccination in order to basically stop the carnage. And I'm giving you my own, this is Sean O'Connell. This is my own feeling about it. But we have now very effective vaccines. We have the ability to make vaccines against the variants. Why shouldn't we put a stop to this and achieve herd immunity as fast as we can through vaccination?
Sean,
Yes,
yeah, this is Joe.
Joe.
Outstanding presentation. I learned a lot. Just to follow up, do you expect that we're going to be getting this COVID vaccination like an annual basis?
I hope not. I hope it's longer than that. This is very hard to predict. I've got to predict what the virus variants are going to do going forward. If we can stop, here's the answer. If we can stop throughout the world, if we can achieve herd immunity in a sizable fraction of the world, this is going to stop the process of variant production. Why? Because you've stopped viral replication. Remember, this bug, the more it can mutate, the more you're liable to get variants. One of the things, I didn't talk about this. We've all heard about super spreader events, but there's something called super mutator events. What these seem to represent are individuals that have a poorer immune system, a not optimal immune system. And when they get the virus, there begins this terrible back and forth battle over months to see whether the virus wins or the patient's immune system wins. In that battle, the virus super mutates. And it's now being thought of, there's a number of studies out that variants like the Brazilian strain, P1, came out of the town of Manaus, and the South African strain, 351, came out of the Cape Town area. It's thought that these arose in super mutator events. That is, people who had an incomplete immune system, an incomplete immune response. I just read a case study of a 42-year-old man, otherwise healthy, but with immune compromised. He was susceptible to immune disorders, and he got the disease, and he fought it for 5-6 months and eventually died. But when they looked at the virus that came out of him at the end of the period, it was massively mutated. And the idea was, it's natural selection within the body. You can either apply this to these unfortunate individuals, or you apply it to a whole population. If you can stop that process, you're going to really slow down, if not stop, the process of variant production. Remember, it's a poor proofreading. It makes mistakes when it replicates. The more you allow it to replicate, the more mistakes, the more likely you're liable to see variants. Influenza, on the other hand, does not cause as severe a disease. And now there are multiple strains. Remember, you got H and N, you got a hemagglutinin and a neuraminidase. There's two proteins on the bug, on the virus, and they can shift, and they shift all over the place. So you have antigen shift and drift. Because of that, it's very complicated. And there's a lot of groups now are working to develop universal vaccines against influenza, including my company. These are vaccines that would not be targeted to the antigens that change every year. Same with COVID. The idea with these new vaccine technologies is to make vaccines that see the whole virus, not just one part, and would be less susceptible to this genetic variation. So two things: you got newer vaccine technologies, as well as achieving herd immunity to slow down the production of variants. I think, Joe, to get back to your case, we're looking at maybe requiring boosters every 3 years or 4 years, but I'm really speculating. I'm sticking my neck way out on this. I don't think, though, it's going to be like flu, where you're going to see it every year. The vaccine technology is drastically improved since the old flu vaccines. It's changed everything.
Yeah. Sean, this is Marge. I have a question. What is
Marge.
the best response to the people I hear saying they will not get the vaccine? I don't know how to speak to these people. What is a quick and
Marge,
simple
And,
response?
and sometimes I have to say, join our club. Some people can listen to reason, and a lot of people just prefer to believe their own reality. We know this, unfortunately, in many fields. It's this whole idea of fake news. Well, anti-vaxxers in many cases is a little bit like that. This is now a will to believe or disbelieve. I think the best way is by example,
Mhm.
but there's a real social imperative here. As we've just been talking about, we need to achieve herd immunity. We need to stop this carnage. And it's one thing to say, I'm not going to wear a mask because that's my own private liberty, but it's another thing to give this virus to other people in the process, to infect people who can't take the vaccine for whatever reason. So my feeling is there's a civic duty, social and civic duty. There is protection of oneself, but there's also protection of one's family and friends. And one of the things I find it is some of these people, you really can't address them with facts. They don't want to listen. So I appreciate, your frustration of this. I find it myself. And I think some of the best things you can do is by example, make sure that everyone around them that is inclined to get vaccinated does get vaccinated.
Mhm.
And the whole idea is that, darn it, shame them into submission. But sometimes you just, I've tried, you can't, I'm a logical guy. Obviously it's kind of what I do. And in many of these cases, that doesn't matter. It doesn't matter. Any amount of facts they can dismiss. And we saw this before COVID, the idea that some of these vaccines caused autism because some of the vaccines had a preservative in it. And, tons of studies show that doesn't happen, but that didn't change their belief. So I think, you try anything that's possible. The one that seems to be the best is do by example. Just say, no, you're wrong. I'm getting vaccinated. My family's getting vaccinated. My company's getting vaccinated. My friends are getting vaccinated. This is the right thing to do because we need to stop. We need to stop this pandemic. We need to stop these deaths and severe disease. That's my best answer. It's a difficult. There's not a good question. There's not a good way to answer those.
Right, thank you.
Anyone else want to chime into that?
Oh, no. Sean, I have a question. I'm Annalisa, Joe's wife.
Yes.
So if someone was diagnosed with COVID in January and they recovered, how long do they have to wait
Good
to get a
question.
vaccine?
2 recommendations on this. First of all, I've heard a lot of people saying, oh, I got to wait 100 days. No, 10 days after their symptoms have gone, 10 days, call it 2 weeks if you want. You're able to get the vaccine. All right.
Oh,
And then here's the second part of it. You only get 1 dose. You've already had the disease. That's your priming dose. So if you've had the disease and you recover from it, you are eligible and should get 1 vaccination after that. And you need to wait about 10 to 14 days after your symptoms have gone. So if you're back to feeling yourself again, and it's been about 2 weeks, you're eligible and you should only take 1. As I said, why? Your disease was the first dose. That's your priming. You basically educated your immune system to the virus. Now the vaccine dose is going to pump that up. It's going to cause more cells, more memory cells to be developed, more different kinds of antibodies. You're just going to have a boosted immune response. Studies have showed that in patients who have recovered from COVID, a second dose doesn't buy you anything. So call it 2 weeks after symptoms are gone and take a single dose. And that includes either the Moderna, the Pfizer, or the J&J, presumably the Novavax. The advice would be the same.
thank you.
Hey,
And then
sure. Oh, I'm sorry.
another question to follow up on that. So
Sure.
if someone has had COVID, but they're a long hauler.
Ah,
I'm hearing a lot more about that too. People had gotten it, and now they're still struggling with various health issues, and they, are deemed like a long hauler. What would
Yeah.
their situation be like?
actually, that gets us into another subject that I could basically teach a whole semester course on, which is the COVID disease. Long hauler, my company, for example, is looking into this long hauling. Long hauling, the long hauler syndrome, if you will, seems to be more a result of the body's response to the virus than the virus itself. All right. It seems to have something to do with the microclotting that the virus causes throughout all the organs. And where that microclotting occurs, that seems to be the affected organ, whether it's heart issues, whether it's cerebral neuro issues, whether it's GI issues, skin issues. There's this microclotting and then the body's response to the microclotting. One of the good news is that vaccination seems to help in many of these patients, the long haulers. They seem to get better, and we don't know why. It seems to now the immune response to the virus was going the wrong direction, causing all these side effects, if you will, that are associated with long haul syndrome. And it's improving it. It's shifting the immune system to just killing the virus and going away and resetting the immune system. And again, I am speculating, but I have been looking at the recent studies about vaccination and long haulers, and they're encouraging. But we're still at the beginning of that. And that's really all I can give you at the moment on that.
Okay, thank you very much.
It's more of overreaction of the body to the viruses, right?
Exactly. I don't know how many here have heard of or know about the 1918-1919 influenza pandemic that was associated with World War I. It's kind of buried in World War I. And I found that our collective memory kind of disappeared. In that epidemic, with the second strain that came back, it seemed the flu originated maybe in somewhere in the Midwest, maybe with the pigs. It went to Europe and it came back. When it came back, it killed young people. This was very unusual. And it killed them. It seemed to kill them two ways. Young people, it killed hours, a few days. Older people, more like the regular flu, would kill them. And it turns out that what happened in the young people was an overwhelmingly abnormal immune response to the virus that would essentially shut down their lungs almost immediately. They essentially drowned. Long haulers with COVID is a little like that, not quite as dramatic, thank the Lord, but it's an abnormal, inappropriate response to the virus. As I said, we think it has something to do with the microclot formation. And that the idea is that with appropriate vaccination, you can reset the immune system so that it does what it's supposed to do and then stops. The problem in long haulers is the immune reaction seems to continue. It continues for months, not longer. So this is why there's a lot of research and these encouraging recent reports that vaccination does seem to play a beneficial, have a beneficial effect in people who are suffering from long haul COVID. Chris, right?
Yes, thank you. I just want to go back. Oh, I'm sorry. Oh, I thought I heard someone else talking. Yeah, just going back to that one point.
No,
You
go
said
ahead.
before. Hello? Okay. About having COVID, recovering from COVID and getting the vaccine. This is a perfect example of my mother. She recovered from COVID in February, probably early February. She got her first shot on this Thursday. Now they scheduled her for a second shot. That's the first I'm hearing about this.
You might want to reconsider.
Okay.
You might want to reconsider if what did she get, the Pfizer?
It's either Pfizer or Moderna.
Or the Moderna, one of the RNAs.
Yeah, it's one of the two because she's scheduled again for April 13th.
Tell me a little bit about her original infection.
Well, she got it. I'm sorry.
Moderate, how, when she got COVID, what was her? How
I'd
did
say
she do?
moderate. She was hospitalized for 3 to 4 days. She went into the hospital on day like 8. They actually gave her medication to take it home, and then her cough was starting to get bad. She went into the hospital on like day 8. She was starting to develop pneumonia. She never really had a drop, like her oxygen level really never went down. Any of those things that they were really worried about. never
Perfect
happened.
candidate. She's a perfect candidate. Moderate disease.
Okay.
And she got over it. She feels good.
Yeah, 100%. It took about, I'd say, a month to get totally back to 100%.
There's nothing wrong with her getting the second vaccine, but the studies are showing that in such patients, they already had a priming dose and a booster, priming dose being her moderate disease, the booster being the first vaccination. She's probably going to not derive much additional benefit as you or I would
Right.
from that second dose. So
Okay.
it's
I'm glad.
optional, but it's in her case probably not necessary.
Wow, thank you so much. That's good to know. I learned something because I'm sure there's other people. That's the first I heard of that, and I know there's a lot of people who've probably had the virus and have been in the process of getting their vaccine. So that might be very useful information. Thank you.
You're quite welcome. Was it Carol that asked? Who?
Yeah, Sean, my question is, will children have to be immunized at some point?
Ah,
And why are they not being immunized, or they're not, on a list?
I would
at
love
all?
to duck and say that's above my pay grade, but that would be a cop out. All right. The short answer is they didn't study kids in the primary studies. They exclude in Pfizer, they went down to 16-year-olds. In Moderna, they went down to 18-year-olds. In J&J, they went to 18-year-olds. But that's a terrible answer. They're now trying to remedy this. So both Pfizer and Moderna and J&J, all three of them, have ongoing studies now in younger populations. And if they have access, John, they have access to this presentation, right? I have slides that basically show, what the current follow-up studies are. One manufacturer, I can't think of it right now. I think it might be Pfizer. They're looking at 6 months to 12 years, and then 13 years to 15 years, something like that. So the idea is they're extending now experience, but I can quote the CDC and I can quote people like Bill Schaffner in Vanderbilt or Tony Fauci. They all said that in order to achieve herd immunity, we are eventually going to have to immunize the kids. That's going to happen. The good news is that we will get a good chunk of the way there, but to get finally to the discussion we had earlier, to stop the disease cold, finally, like we did with polio, we're going to probably have to immunize most of the kids. And so in order to do that, we need to know what the right dosage is. How does the vaccine work in them compared to others? And a similar question I get is, well, what about women who are pregnant or thinking about being pregnant, or women who are breastfeeding? The answer is yes, absolutely get vaccinated. There's a gal, Dr. Elizabeth Miller. She's head of obstetrics and gynecology in Northwestern. And she came up with an amazing op-ed piece in New England Journal of Medicine, where she reviewed in all the pivotal trials, there were women who became pregnant during the trials and then gave birth after the trials. And there was no evidence whatsoever of adverse events, no effect on the pregnancy. Not only that, what they were able to show you that in the nursing mothers, the protective antibodies would cross into the milk and confer protection on the kids. So in the case of, I refer to Dr. Miller, but from her strong opinion, and Dr. Wen in Baltimore said the same thing. These are all mothers who have had the vaccines. They said, get the vaccine, get the vaccine. In the kids, we need to test. We do need to test because in the children in particular, vaccines can work differently. We know this with pharmaceuticals in general, drugs in general. You can't extrapolate adults to children and just reduce the dosage. You really need to see how it works. Well, the good news is we're doing that. We're doing that. And these studies are ongoing. You know, each one of these studies has several thousand, up to 7,000, I think, in one case. So we're going to have answers to the vaccine usage in the kids by the summer, I think. But in pregnancy, you know, Dr. Miller and Dr. Wen, they made believers out of me. In pregnancy, women undergo a selective immunosuppression. And one of the things, unfortunately, we do know from this terrible pandemic is it hits pregnant women real hard. So my feeling is, you know, going with Dr. Miller, going with Dr. Wen, Dr. Grounder, who is on Biden's task force, she runs it, they should get the vaccine.
Thank you.
Any other questions? We're doing good. We're rolling good. We're at the minute 10 mark. We got time for a few more questions. Pastor, you tell us when we have to stop.
Sure, yeah. Any other questions you want to ask Sean? Steve?
I have one question. So mine was kind of like Carol, wondering if we could get to herd immunity without our children, and we can't. So at this point, like my children are all 16 and up, so they are considered safe just to get
Yeah,
them
get the vaccine, yeah.
and just do it. And you have no hesitation on that for teenagers because of the studies?
Not if they're 16 or older, no. There was nothing showed up. Nothing, there was no blips, no increases in side effects, no increase, no lack of efficacy, all the tripwires, nothing showed up. They responded in the Pfizer 16 and above just fine. They responded in the Moderna 18 up. The one thing you do see in the kids, younger adults and older teenagers, is in the second dose, they might have more transient side effects. They might get a mild fever. They might get a bad headache. They might get chills, shaking, body aches, but nothing that a couple ibuprofen, a couple Advils won't cover. And the reason is they have stronger immune systems than we do. It's very simple. So when they get that second dose, they might get hit a little harder than we get. I had my second dose, pardon me, I'm in my later 60s. I didn't even notice it. Little thing on the arm, all right, fine. But I've seen some younger people, a couple of my associates, clinician associates who were in their late 30s, they got hit real hard by the second one. They got hit hard. But so you might expect that, but these side effects don't last 24 hours and they're gone.
But unfortunately, long term, we don't know if it's going to affect anything else with the young kids.
We don't know anything long term, given that the first immunizations occurred in December. Our past president, he got it then. We don't know. We're looking, but so far, knock on wood, millions of patients have been treated and nothing is showing up yet. But that's why I said they always do post-approval surveillance. You have to keep an eye out for this thing. But so far, we're looking good. We're looking very good.
So Joe and I can attest to the Moderna shot because we both had our second dose. And the first dose, I was fine. I didn't have any issues. He had the chills and the headache. I had a slight headache the next day and we both took Tylenol and we were fine.
And it also says that your immune systems are up to snuff.
Yeah. So, but the second dose, he was fine, and I'm the one who had the chills and the headache. But again, we both took the Tylenol, and we were ready for that. And so, and the worst of it was just like the slight headache, but other than that, we were fine. Like within 24 hours, it was gone.
Excellent.
Yeah. And but anyway, that's.
Yeah, my mom had after the first shot, she had slight symptoms for yesterday. She had the chills, a little fever.
And you would expect that because you're boosting her immune system. She's already gotten her initial dose.
Right.
Disease itself. And now what you've just done is you've just made her immune system say, oh, here it is again. And
yeah, right.
Jumps into action. And, yeah, this is not unexpected. So I'm glad. And as I said before, she may not need that second shot.
Now, with that, speaking to that again, I'm sorry to monopolize the time. I have other family members who have recovered like way earlier, but that's still, you think that would still apply, like earlier back in November?
Way earlier is now measured in months, not years.
Right, like say they like had it in like November. When they get in line for the vaccine, should they still take the 2 shots, or you think that's
It's up to them. They need the 1 shot for sure.
Okay.
Whether they get the second shot or not, that's up to them.
okay?
Well, it's not going to hurt them. The
Right,
question
right.
is, how much benefit is it going to give them?
Interesting. Thank you.
Hi, Sean. I've heard this new mRNA process is going to be used for other things. Like I've heard that they think it might be useful for curing cancer and things like that. Do you know anything about that you could talk to?
We personally, and I'm going to speak off track. I hope my people aren't watching. We are interested in the viral vector approach. But the RNA approach certainly is a good one. And in the case of cancers, it could make a lot of sense. The idea is it's easier to make and it's easy to administer. It's harder to store. RNA is pretty fragile stuff. When I studied it in school, we had a big sign in our lab say, beware the finger aces. What does that mean? It means even on your hands, you made enzymes that would eat RNA. RNA, you had to really fight to keep it intact. So they're fragile. That's why they store the Pfizer at -80 and the Moderna at -20. But in the case of cancer, that's a specialized issue. And it could be that your cancer therapies could be delivered quicker. What we're working on is we're working on cell therapies, and we don't like adenovirus. We like vaccinia, the old pox virus, because it's a big virus. You can do lots with it, and it's very immunogenic. So we use vaccinia to get a good immune response, or we use whole cells themselves, either from donors or patients. But yes, to your point, if it works this well in this case, why doesn't it work well for, let's say, melanoma? Or why doesn't it work for adeno breast cancer, something like that? And yes, I'm sure they're going to use it for those applications. Anything else?
I have a question.
Betty.
I'm from a church in Central Jersey.
Mhm.
My question is that if you take the vaccine, are there any preventive things you should or should not do to minimize the symptoms? I was an unfortunate victim of the fever and all the aches. They told me to drink water. I was careful about not using painkillers, although I had with the first shot, I couldn't even lift my arm. It was so sore for just 24 hours. But are there things that people can or should or should not do to minimize side effects?
Yeah, there's a couple things you can do. One, drink lots of water and fluids.
Okay.
1, 2, get a lot of sleep. Be rested. All right. And with respect to analgesics, Tylenol is the easiest. If that works for you, absolutely. Acetaminophen is by far the easiest. I have no problem with ibuprofen, Advil, and the like. I wouldn't use anything stronger. I would stay away from aspirins.
Mhm.
I would stay away from the more potent non-steroidals. I wouldn't use naproxen, believe. I would stay with either Tylenol, and if Tylenol is not strong enough, Advil, ibuprofen, generic ibuprofen seems to work fine. There's no interaction between those compounds and the vaccine that anyone's described. But as I said, you want to make sure that you're well hydrated and you're well rested, and you do have those simple pain meds available. That's
Okay, thank you.
Oh, and one other thing, I'd probably stay away from alcohol. Stay away from alcohol. So if you like the glass of wine at dinner, you might want to forego that for the night before and the night after you get the infection, or the injections. Anything else? Very good questions. Group, very good questions indeed. Well, again, I thank you for taking the time to listen to me. And if you have any additional questions, you can funnel them through the pastor or through John, and I will get right back to you.
Well,
Thank you very much. That was wonderful.
This was a very fascinating, informing presentation. We really appreciate you sharing expertise, knowledge about vaccines, and may God continue to bless you as you
And may God bless you all too.
research and inform the public about the vaccines.
Very good. Everyone, please enjoy their weekend. Stay safe.
Yes, yes.
You too. Thank you.
Could
Bye.
we end with a word of prayer? Just a short.
Absolutely.
Let us pray. Loving, gracious God, we give you thanks and praise for this beautiful day. And we thank you for this opportunity to come together as a church family and learn about COVID-19 vaccines. Lord, it has been a very challenging year, very, very challenging year. And we trust that you have faithfully guided us, led us. And we are grateful to you, Lord, for the light beginning to emerge at the end of a long, deadly tunnel. We are grateful for all those tireless labors of researchers and scientists who toiled day and night. And we thank you, Lord, for all the work of doctors and nurses and healthcare workers,
Amen.
officials, and people like Sean who work hard to share the vaccines and share the knowledge about the vaccines the vaccines that restore our community to wholeness and health. Grant them your strength and peace, endurance and wisdom. We pray for all who are desperately ill, for those who grieve the loss of their loved ones during this time. We ask for your mercy. We ask for your blessings, oh God. Again, we are grateful for the work of Sean and his presentation. We ask for your continued blessings upon Sean and his work, and blessings upon our church. We pray all this in the name of Jesus Christ.
Amen. Amen.
Okay.
Amen.
Thank you, Sean, again, and thank you everybody
Pastor,
for your
Thank you.
excellent questions and participation today.
my pleasure. The pleasure was mine, Pastor.
Thank you, Sean.
Everyone have a wonderful weekend. Take care.
Bye.
You too. Thank you.
Thank you.
Okay, thanks.